Alright, let's dive into something that's both mind-blowing and potentially game-changing in the world of cancer research. Imagine this, a new therapy that could outsmart one of the trickiest forms of liver cancer. Sounds like sci-fi, right? But it's not. It's happening right now in labs, and it's all about something called ferroptosis and how we can use it to fight back against hepatocellular carcinoma, HCC. Personally, I think this is one of the most exciting developments in cancer research in recent years, and here's why. First, let's set the stage. HCC is a brutal disease, often driven by mutations in a protein called catenin. What makes this really interesting is that these mutations don't just cause cancer, they also make it resistant to treatment. One of the key players in this resistance is the mTOR pathway, which, when overactive, suppresses a type of cell death called ferroptosis. Ferroptosis is like a self-destruct button for cancer cells, but many tumors find ways to disable it. So, if we can reactivate ferroptosis, we might have a powerful weapon against HCC. Now, here's where it gets fascinating. Researchers have been experimenting with combination therapies to overcome this resistance. They're not just throwing drugs at the problem, they're strategically targeting multiple pathways at once. For instance, they're using mTOR inhibitors like rapamycin alongside ERK inhibitors like PD901. What many people don't realize is that these pathways don't work in isolation, they're part of a complex network. By hitting them simultaneously, the therapy doesn't just kill cancer cells, it also prevents the tumor from finding a workaround. One thing that immediately stands out is the role of a protein called EBP1. In its mutated form, for EBP1A4, it actually promotes ferroptosis and makes the cancer cells more sensitive to treatment. How? It competes with another protein, KEAP1, to bind to HSP90-beta. This might sound like molecular trivia, but it's huge. By displacing KEAP1, 4-EBP1A4 allows for the degradation of NRF2, a protein that normally protects cells from ferroptosis. If you take a step back and think about it, this is like sabotaging the cancer cell's defense system from the inside. What this really suggests is that we're not just treating cancer, we're outsmarting it. The combination therapy doesn't just suppress tumor growth, it creates a hostile environment where the cancer cells can't adapt. In my opinion, this is the future of cancer treatment, personalized, multi-targeted, and designed to exploit the tumor's weaknesses. It's not just about killing cancer cells, it's about making sure they can't come back. But here's the bigger question. What does this mean for patients? If this therapy makes it to clinical trials and proves effective, it could be a game-changer for people with catenin-mutant HCC. Right now, their options are limited, and the prognosis is often grim. This raises a deeper question. How quickly can we translate these findings into real-world treatments? And how can we ensure that they're accessible to everyone who needs them? A detail I find fascinating is how this research connects to broader trends in cancer biology. We're moving away from one-size-fits-all treatments toward therapies that target specific mutations and pathways. This isn't just about HCC, it's about a new paradigm in cancer research. From my perspective, this study is a perfect example of how deep molecular understanding can lead to innovative treatments. So, where does this leave us? Personally, I think this is just the beginning. We're scratching the surface of what's possible with combination therapies and ferroptosis induction. The implications are massive, not just for liver cancer but for other cancers that rely on similar pathways. What makes this research so compelling is that it's not just about finding a new drug, it's about understanding how cancer cells think and then beating them at their own game. As we wrap up, I'll leave you with this thought. Cancer is clever, but science is relentless. Studies like this remind us that even the most stubborn diseases have weaknesses. The question is, how far are we willing to go to exploit them? Let me know what you think in the comments. Is this the future of cancer treatment, or is there another approach you're excited about? Let's keep the conversation going.